So in the past couple of days, we've been talking about mitosis. And mitosis was the division of somatic cells or body cells. It's what we do when we grow and develop. It's what we do when we repair damaged tissue. It's how things like bacteria reproduce. And it's linked to the cell cycle, and when that goes wrong, it's linked to diseases like cancer. Today we're going to be talking about something different. We're going to be talking about a process called meiosis. And meiosis is the division of the nucleus and genetic material that results in sex cells. Gametes is what we call them. Okay, so today we're going to be talking about this process specific to sex cells or gametes. We're no longer talking about body cells. And so some examples of these that you might know of are sperm, egg, and pollen in plants. Okay? This is a really important process. This is a process that allows for sexual reproduction and results in a lot of genetic diversity or differences amongst living things. In humans, for men, for boys, this process occurs at puberty. Okay, we don't have the ability to start making sex cells, sperm and egg, before this time. For girls, it's a completely different story. This actually happens prior to birth and in the first years of life. After that time, you no longer make any more eggs. Okay? So men, after puberty, you'll make them for the rest of your life. For girls, after the first years, you will no longer make any more. This is important for us to know when we start talking about things like fertility. Okay? So your doctor definitely wants to know about this. Again, meiosis creates sex cells, or what we call gametes. And there are some major differences in this process. Okay, so there are a couple different ways that this is different from mitosis. One way that this process is different from mitosis is that it results in actually 4 cells, and these cells are genetically diverse from one another. Remember, in mitosis, we made 2 genetically identical cells. Here we're making 4 genetically diverse cells. Major difference. These cells are also what we call haploid. The prefix hap means half. Okay, in these cells, the result is 4 cells that have 1 copy of each chromosome. One copy of each chromosome. 1 from mom, or 1 from dad. This word "or" is key. When we were talking about mitosis, we used the word "and." We were making diploid cells, cells that had 2 copies of each chromosome. We got 1 from mom and 1 from dad. In meiosis, we're making haploid cells. You either get 1 from mom or 1 from dad. This is one source of that genetic diversity. There's a reason this is important. And I'm sure you might know what it is. It allows for sexual reproduction. Okay? It allows for a sperm with one copy of a chromosome to combine with an egg with one copy of each chromosome. Okay, so it's important because it allows for sexual reproduction and for sperm and egg to combine with one another. The end result, that should say result, is a diploid zygote. All zygote is a fancy word for the first cell that gets made in sexual reproduction, for a fertilized egg. Every single one of you in this room started out as a zygote. Okay. Go ahead, and I'm going to have you pause the video after I say this. Give the students 1 minute to analyze this diagram over here and look at the notes above to answer this question. How is meiosis different from mitosis? Okay, so stop the video, give the students 1 minute to go ahead and answer this question, discuss amongst themselves. Come up with a list, and then we'll talk about this list. Okay, one way it's different is we end up with one copy of each chromosome in our resulting cells. Another way it's different is all cells are genetically diverse. They're different from one another. In mitosis, they were all the same. We make 4 cells. If you looked at the diagram closely, there are 2 divisions in meiosis. Our cells divide twice. And here we're talking about sex cells and sexual reproduction. We're no longer talking about body cells or somatic cells. You may need to stop the video to just review this with the students and go through it one more time to make sure that they have got everything that they need. Okay. This is a diagram that we've seen many times now. It represents the cell cycle. Okay. The cell cycle for meiosis is really no different than the cell cycle from mitosis. The cell still needs to grow and mature. And before it can move on, it still needs to pass some major checkpoints. After it passes the checkpoint, we can still do DNA replication. We still need to make more copies of our DNA. And after that, we still need to make sure that that happened correctly. So we still have another checkpoint. And our cell will still do some more growing after the replication of our DNA. The only difference is we're no longer talking about mitosis here. We're now talking about meiosis. Okay. The other difference is we said that there was 2 divisions. So we don't go through this division phase just one time. We actually go through this 2 times. Okay. So the cell is still spending the majority of its lifetime in interphase. And during interphase, the cell is still growing and replicating its DNA. Even though we go through 2 division processes, it only enters interphase 1 time. And it goes through 2 division cycles. Okay. All right, so we can see some similarities. We can see some major differences here. Let's now look very closely at the different phases of meiosis. Okay. The first phase is prophase. Enters prophase with the nucleus, and we can't see the genetic material. It hasn't condensed yet, but over time, our nucleus will begin to dissolve, and our genetic material will become more visible. So our genetic material begins to condense, and the nucleus begins to dissolve. Okay. Very similar to prophase of mitosis. Now, metaphase, this is where we see our major difference. Major difference and our source of diversity. In mitosis, in metaphase, our chromosomes lined up like this down the center of the cell. Okay. In metaphase, in meiosis, our spindle fibers are still attaching to our chromosomes, and they're still pushing and pulling to line up our chromosomes at the center of the cell. Except for my alike chromosomes, my homologous chromosomes line up side by side. Again, our chromosomes that are similar to one another are called homologous chromosomes. The prefix homo means same. So my chromosomes that have the same genetic material are lining up side by side. Okay. The cell produces a string-like proteins called spindle fibers. And the spindle fibers attach to the center of the chromosome, just like in metaphase of mitosis. And the spindle fibers pull the chromosome to the center of the cell. This is an important step to make sure that each new cell gets the correct amount of genetic material. Again, this checkpoint here at this point is very important to ensure that. All right. Our other key piece was that our homologous chromosomes line up side by side. Okay. Now there are some major differences in metaphase of meiosis. One of those major differences is crossing over. Okay. Crossing over is one of the major sources of diversity. It's a major source of diversity here. Okay. What's happening is my homologous chromosomes do not want to lose each other. They know they have to line up side by side. So in order to make sure that they stay together, they intertwine. So imagine my fingers being chromosomes. They're going to interlock with one another. The arms of them are going to intertwine. Okay? Just like if you go to a busy mall and you don't want to lose your significant other, you're going to hold their hand. Okay? Same idea. But sometimes when these guys intertwine with one another, they swap genetic material. And so part of my red chromosome swaps to my blue one, and part of my blue chromosome swaps to my red one. I've swapped genetic material, I've created some differences. One source of genetic diversity is crossing over. A second source is this idea of independent assortment. This is a fancy way of saying that my homologous chromosomes can line up any which way that they choose. Okay? So if let's see here, I gotta find some different colored pens and pencils. All right? So let's just take a look. All this is saying is that, let's say these two chromosomes code for eye color. And these two chromosomes have the genes that code for height. Okay? These are homologous chromosomes. When these cells separate, I'm going to end up with a black and a red over here, and a purple and a blue over here. But what if they lined up differently? What if instead of having black over here and purple over here, I put purple on this side and black on this side, and I kept the bottom ones the same, red and blue. My homologous chromosomes are still side by side, so I'm still all good there. But when they separate, I'm going to end up with this. I'm going to end up with purple over here and red. That's different from up here, which was black and red. And over here, I'm going to end up with black and blue, which was different than up here where I had purple and blue. So depending on how my chromosomes line up, I could end up with differences in my cells. The combinations of the genes that are there are going to be different. Another second source of genetic diversity. The next phase is very similar to what we've seen in mitosis. It's anaphase. My spindle fibers begin to shorten, and my homologous chromosomes are separated from one another. So spindle fibers shorten, and the chromosomes are pulled to the ends of the cell. But here's the key part. My homologous chromosomes are separated. In telophase, all that's happening is I'm starting to see the actual division of the cells. That cleavage furrow is beginning to form. Okay. Notice my nucleus does actually not reform here. But I'm just starting to see two new cells are actually beginning to form here. And then to end it all off, my first division of two, I go through cytokinesis, which meant cell movement. My cells actually divide. A protein squeezes the cell membranes together until they split. So now I have two genetically diverse but diploid cells. Remember, diploid is not what I want. Okay? I want to make haploid cells, so I have to go through a second division process. That second division process is almost identical to mitosis. There's no special tricks here. In metaphase, there's no, we have no crossing over. I'm just going to abbreviate that CO. And there's no independent assortment. I'm just going to abbreviate that IA. Okay? So there's no special things going on here. This is very similar to mitosis in that way. Okay? If you want to review these two diagrams on your own, this is going to show you specifically how sperm are made, and this is going to show you more specifically how eggs are made. Okay, so you can review those on your own. But what you're going to notice is that in eggs, there's these things called polar bodies that get formed. Okay, of the 4 cells, only actually one of them is viable, meaning one of them actually works when we're making eggs, when females are making eggs. Okay. The other 3 are too small. They're called polar bodies, and they actually end up dying off. In males, all 4 of them work. Okay. In the end, meiosis allows for sexual reproduction, and it's important because it creates diversity. And diversity over many, many generations allows for adaptation to our environment and even evolution over time. Now, this process doesn't always happen perfectly. Sometimes our homologous chromosomes do not spread apart or do not separate. That's called non-disjunction. So, homologous chromosomes don't separate. Okay? And so we end up with 2 cells that don't have the right amount of genetic information. They have too little. Those guys typically don't survive. And we have 2 cells that have too much genetic information. Those guys actually typically will survive. But when they combine with a sperm or an egg, the resulting cell has too many chromosomes. And this can result in something like Down syndrome. Okay. You may need to go back and review some of this stuff. And you also are going to want to review pages 44 to 50 in your iBooks. It will review all of these concepts with you and should make them a little bit more solidified. Okay? Thank you very much, and have a great afternoon.